Hello everybody, I am Dr. Sumati from the Department of Biochemistry, Biotechnology and Bioinformatics. I am dealing with modules under Nutritional Biochemistry. We have seen already about the classification and some concepts about the metabolism of carbohydrates. Now, in this present module, we will be seeing about glycogen metabolism and HEXOSE monophosphate shunt. The major site of daily glucose consumption, that is nearly 75%, is the brain. Most of the remainder is used by erythrocytes, heart muscle and skeletal muscle. The body gets glucose either directly from the diet or from amino acids and lactate via a process called gluconeogenesis. Glucose that is got from these two primary sources either remains soluble in the body fluids or it may be stored in the polymeric form, namely glycogen. Now, this glycogen is considered the main storage form of glucose and is found mostly in the liver and in the muscle. The liver is the major storage organ in the form of glycogen. Muscle has much lower quantity of glycogen per unit mass of tissue, but since the total mass of muscle is so much greater than that of liver, the total glycogen stored in the muscle is about twice that of the liver. Moreover, the kidney and the intestine adds minor storage sites. This glycogen storage in liver are considered the main buffer of blood glucose levels. So, understanding the mechanisms by which glycogen helps to maintain the blood glucose level becomes important. We have already discussed in our previous modules how ATP, which fuels many metabolic processes, is synthesized by the electron transport chain. Now, there is yet another pathway by which this NADPH, which is an important reducing equivalent, is synthesized, and that is called the HEXOSE monophosphate shunt. The biosynthetic reactions such as cholesterol synthesis, fatty acid synthesis, drug reduction, etc. requires NADPH. So, the HEXOSE monophosphate shunt pathway also permits the entry of some carbohydrates into the glycolytic pathway. Most important is ribose, but also some other sugars are there, and hence it acts as a connection route between different pathways. So, the objectives of this module is to give an overview of glycogen metabolism, to understand the factors that control the glycogen metabolism by intracellular signaling, and to understand the function of the pentose phosphate pathway in the production of NADPH and ribose precursor for nucleic acid synthesis. Now, we know that glycogen is an animal storage form of branched polymer of glucose. All cells contain glycogen, but it is most prevalent in the liver and in the muscles. Glycogen homeostasis involves the concerted regulation of the rate of glycogen synthesis called as glycogenesis and the rate of glycogen breakdown called glycogenolysis. These two processes are reciprocally regulated in such that the hormones that stimulate glycogenolysis, that is glucagon, cortisol, epinephrine, norepinephrine, at the same time inhibit glucogenesis. Conversely, insulin, which directs the body to store excess carbon for future use, stimulates glycogenesis while concurrently inhibiting glycogenolysis. About the regulation by hormones, we shall discuss in the next module. The HEXOSE monophosphate pathway is used for the production of NADPH from NADP. The biosynthetic reactions such as cholesterol synthesis, fatty acid synthesis, drug reduction requires NADPH. It also acts as a cofactor for some non-synthetic enzymatic reactions. Furthermore, it is also used for the production of ribose for nucleotide and nucleic acid synthesis. So, the HEXOSE monophosphate pathway also permits the entry of some carbohydrates into the glycolytic pathway, especially ribose, but also some other sugars, and hence acts as a connection route between different pathways. Now, why is this glycogen metabolism considered to be so vital? It is for several reasons. Glycogen stores in the liver are used to maintain a constant blood glucose concentration. Glycogen storage is also maintained by muscles as a reservoir of glucose for strenuous muscular activity. The synthesis and degradation of glycogen takes place by different metabolic pathways, allowing for reciprocal regulation. In addition, the enzymes of glycogen metabolism are under hormonal regulation. The biochemical pioneers of glycogen metabolism were the Corys, that is Carl Corey and Gerty Coley, a husband and wife team. They demonstrated that glycogen is broken down by phosphorylases. The complete breakdown process of glycogen is glycogen will be broken down to glucose 1-phosphate and one residue of glucose minus 1. Process of glycogen synthesis is the reverse of it, where subsequently one molecule of glucose will be added to the glycogen chain. Both the processes, namely glycogen breakdown and synthesis, is controlled by two key enzymes, which are glycogen phosphorylase and glycogen synthase. The activities which are activated or inactivated by allosteric regulation and phosphorylation or dephosphorylation. The storage form of glucose in most eukaryotic cells except plants is glycogen, and we are aware about the structure of glycogen, which is large highly branched polysaccharide containing glucose units joined by both alpha 1,4 and alpha 1,6 glycosidic bonds, and it is made up of both amylose and amylopectin. And again, the liver and muscles store glycogen and hence have the necessary anabolic and catabolic enzyme. The degradation and synthesis of glycogen occurs in the cytosol, and the substrate for these reactions is the free ends of the branched polymer. The large number of branch points in the glycogen results in the generation of multiple non-reducing ends that provide a highly efficient mechanism to quickly release and store glucose. First, let us see the steps involved in glycogen synthesis. It was scientist Louis Leloir who discovered that glycogen biosynthetic pathway. It can be in simple terms said as glycogen combines with UDP glucose to give glycogen N+1 and UDP. So, to start with, UDP glucose is formed from glucose by the addition of UDP by the enzyme glucose pyrophosphorylase. Then, the glycosidic bond between glucose and UDP in UDP glucose is hydrolyzed. In the next step, the glucose unit of UDP glucose is transferred to the C4 hydroxyl group on one of the glycogen's non-reducing ends to form a alpha 1,4 glycosidic bond, and the reaction is catalyzed by the enzyme glycogen synthase, which forms an alpha 1,4 linkage, and sequential glucose residues are added. As we are aware that glycogen is a highly branched polymer, this enzyme, that is glycogen synthase, can only synthesize a linear polymer. Now, another important enzyme comes into play. It is called the branching enzyme or amylo alpha 1,4 alpha 1,6 trans glycosylase. Now, this branching enzyme takes a block of 7 or so residues from the non-reducing end and transfers these 7 residues to the interior site and creates an alpha 1,6 linkage. The chain that contributes the 7 residues must be at least 11 residues long, and the new branch point must be at least 4 residues away from the pre-existing branch points. About 7 units of the non-reducing end of alpha amylose chain are removed at the alpha 1,4 linkage and reattached to the C6 of alpha amylose chain by alpha 1,6 linkage. Thus, the chain gets elongated and branched. So, whenever glucose is needed, glycogen serves as a ready reserve and breaks down by a process called as glycogenolysis. This picture shows the reaction sequence. First, glycogen phosphorylase catalyzes the successive phosphorylases of glucose residues starting from the non-reducing end. The phosphorylated glucose cannot diffuse out of the cell. Now, here again this enzyme cannot act at the branch point. So, it stops until it gets 4 residues away from the alpha 1,6 linkage where you have the branch point. Now, another enzyme is required to remove these branches, and this enzyme is called as the debranching enzyme. This enzyme releases a glucose unit one by one until it reaches approximately 5 units from the branch point. There are 2 forms of this enzyme phosphorylase, namely phosphorylase A, which has a phosphate attachment, and phosphorylase B, which has no phosphate attachment. Now, the reaction mechanism includes cleavage of the glycosidic bond by acid catalysis to form glucose 1-phosphate. Then another enzyme called as alpha 1,6 glycosidase cleaves the alpha 1,6 glycosidic linkage, after which the glycogen debranching enzyme removes branches so that glycogen phosphorylase can complete the reaction. There is an enzyme called phosphoglucokinase which phosphorylates glucose 1-phosphate to form glucose 1,6 bisphosphate. It would be nice if this intermediate can be converted to something which will enter into glycolysis. This is done by the enzyme phosphoglucomutase that converts glucose 1-phosphate produced from glycogen breakdown to glucose 6-phosphate in order to enter glycolysis or to produce glucose in liver. 90% of glycogen is phosphorylatically cleaved into glucose 1-phosphate, which is then isomerized to glucose 6-phosphate, whereas 10% are the branched residues which are hydrolyzed into glucose, which can be phosphorylated into glucose 6-phosphate. The complete oxidation of glucose 6-phosphate through glycolysis, the citric acid cycle and oxidative phosphorylation yields 38 molecules of ATP. So, the overall efficiency of storage is 97%. So, the entire process of glycogen metabolism is regulated by 3 processes. One is by allosteric regulation of glycogen synthase and glycogen phosphorylase. 2 by hormonal control of glycogen metabolism, and finally 3 by regulated differences between liver and muscle phosphorylase. First, let us see about the allosteric regulation of glycogen synthase and phosphorylase. Look at this figure. Whenever ATP is exhausted, it is an excellent reason to release glucose from the store in order to make some more. The glucose 6-phosphate ATP and AMP allosterically regulate the activity of the two enzymes glycogen phosphorylase and glycogen synthase. On the other hand, glucose 6-phosphate will be plentiful when glucose itself is abundant and therefore signals an opportunity for replenishing the glycogen stores. Next, how hormonal control of glycogen metabolism regulates the glycogen synthesis and breakdown. This we shall discuss in detail in the next module. The key fact you should know is insulin, which is an anabolic hormone and promotes the storage of fuels. Glucagon is a hormone that promotes the mobilization of fuels. All of glucagon's effects are opposed by effects of insulin. So, if stimulation of insulin takes place, it releases and suppresses the release of glucagon. The third mechanism is by regulatory differences between the liver and muscle phosphorylase. There are regulatory differences between glycogen phosphorylase in the muscle and in the liver. The liver enzyme is inhibited by glucose, but this does not inhibit the muscle enzyme. Calcium stimulates the muscle enzyme, but not the liver enzyme. Recall that calcium is also a trigger for muscular contraction. The simultaneous stimulation of glycogen breakdown therefore anticipates an increased demand for ATP. Again, there is also an inter-organ relationship in glycogen metabolism. They are liver glycogen utilization, muscle glycogen utilization and the Coricycle. The two tissues that have most significant pools of glycogen are the liver and the skeletal muscles. Liver glycogen is turned over quickly. It serves as a major reserve of blood glucose during short-term fasts. After liver glycogen is depleted, muscle glycogen can be drawn down. This, however, requires some roundabout metabolic trickery. Now, coming to liver glycogen utilization. The liver mobilizes glucose from its glycogen store. This is transported to the endoplasmic reticulum where it is dephosphorylated. Glucose is then taken back to the cytosol and released into the bloodstream. Now, some of the glucose will be rephosphorylated before making it out of the cell, creating a futile cycle. However, the dominant glucose phosphorylating enzyme in the liver is glucokinase. This enzyme has fairly low affinity for glucose. Therefore, enough glucose will escape rephosphorylation and be released into the bloodstream. Coming to muscle glycogen utilization, muscle glycogen primarily serves the energy needs of the muscle tissue itself. During prolonged physical exercise, maximum of it is broken down to glucose 6-phosphate and then used up by the usual pathways right within the cells that stored it. Under suitable conditions, that is prolonged fast without physical exercise, muscle glycogen can also contribute to the replenishment of blood glucose and therefore will keep the intracellular level of free glucose well below the extracellular concentration. The net transport of glucose should therefore be directed inwards all times. Next, coming to the Coricycle. While skeletal muscle gets glucose by oxidative metabolism most of the time, some other tissues, especially the red blood cells and the lymphocytes, depend mostly or even exclusively on anaerobic glycolysis even under aerobic conditions. This peripheral tissue releases lactate and it is scooped up by the liver, which converts it back into glucose through gluconeogenesis. This process is called as Coricycle, named after the discoverers Carl and Gerty Cori, who worked it out as early as 1929. This again we will discuss in detail in the next module. Now, we will move on to the next topic, that is the HEXOSE monophosphate shunt. The HEXOSE monophosphate pathway, otherwise called as the pentose phosphate pathway, is involved in the production of NADPH required for a variety of cellular functions. This NADPH production is controlled by the relative levels of NADP and NADPH that alter the activity of one important enzyme, that is glucose 6-phosphate dehydrogenase. This is the control enzyme for the pathway. The ribose 5-phosphate required for nucleotide and nucleic acid biosynthesis is also synthesized by this HMP pathway. NADPH is part of the mechanism that red blood cells use to prevent oxygen mediated damage to their membrane and proteins and to maintain hemoglobin in the oxidation state required for the oxygen transport. Now, let us have an overview of the shunt pathway. We can break this pathway into two parts, namely oxidative pathway and non-oxidative pathway. In the oxidative phase, glucose 6-phosphate is oxidized with the generation of 2 molecules of NADPH and 1 molecule of pentose phosphate with the release of 1 molecule of carbon dioxide. Now, what happens in the non-oxidative phase is that the pentose phosphate is transformed to intermediates of glycolysis. Again, let us see the stage 1 of the oxidative phase. This figure shows the steps involved. In step 1, glucose 6-phosphate is oxidized by NADP plus to produce 6-phosphoglucanolactone. In this reaction, 1 molecule of NADPH is formed and regulation is effected by glucose 6-phosphate dehydrogenase enzyme. In step 2, this lactone is hydrolyzed by an enzyme lactonase to form 6-phosphogluconate, which is a linear molecule. And in step 3, which is an oxidative step, together with decarboxylation, the enzyme 6-phosphogluconate dehydrogenase will oxidize alcohol to a carbonyl and decarboxylation to give ribulose 5-phosphate, and a second molecule of NADPH is generated. Now, coming to the non-oxidative phase, here again we see the steps involved are, the first step will be isomerization, that is the ribulose 5-phosphate will be isomerized to ribose 5-phosphate or may be epimerized to xylulose 5-phosphate. Next is a trans-ketolase reaction which transfers 2 carbon unit with a keto group from the xylulose 5-phosphate to ribose 5-phosphate to form a 3 carbon, that is glyceraldehyde 3-phosphate, and a 7 carbon sugar which is pseudoheptulose 7-phosphate. In the next reaction catalyzed by trans-aldolase, it involves the transfer of a 3 carbon unit from the 7 carbon, that is pseudoheptulose 7-phosphate, to glyceraldehyde 3-phosphate to form a 6 carbon, that is fructose 6-phosphate. There is another trans-ketolase reaction. Here again a 2 carbon unit is transferred from xylulose 5-phosphate to erythrose 4-phosphate to form 6 carbon sugar, namely fructose 6-phosphate and glyceraldehyde 3-phosphate. Now, the next step is the regeneration of glucose 6-phosphate. Now, 2 molecules of glyceraldehyde 3-phosphate formed in step 7 are condensed to form 1 fructose 6-phosphate. So, this fructose 6-phosphate will then be converted to glucose 6-phosphate, which will be nothing but the reversal of step 2 of glycolysis. Now, what do you think is the significance of this pathway? Firstly, this pentose phosphate pathway protects the cells against reactive oxygen species, which includes molecular oxygen and partially reduced reactive forms of oxygen like superoxide, hydrogen peroxide, hydroxyl radical, etc., which forms a reactive oxygen species. Second, the NADPH and glutathione that protects the cells against highly reactive oxygen derivatives are generated. The cells are protected by reduced glutathione by destroying the hydrogen peroxide and hydroxyl free radicals. The generation of glutathione from its oxidized form requires the NADPH that is produced in the glucose 6-phosphate dehydrogenase reaction. Next, we will see the importance of this pathway in RBCs. The role of red blood cells in oxygen transport makes them liable for oxidative damage by H2O2. Now, this hydrogen peroxide causes both oxidation of iron in hemoglobin to form methemoglobin, and it also causes lipid peroxidation of the RBC membrane. The production of NADPH is a major role of the HMP shunt in the red blood cells, so that it protects these cells from oxidative damage by providing reduced glutathione for the removal of dangerous hydrogen peroxide. Chemically, NAD and NADP are very similar molecules. These two molecules are structurally closely identical. The catabolic processes such as TCA cycle, NAD is used to accept electrons, whereas NADPH is primarily used to donate electrons for synthetic reactions. NADPH can thus act as a strong driving force for otherwise unfavorable reactions. Variety of electron donation reactions uses NADPH. In some cell types, NADPH is required to maintain normal functioning in addition to its involvement in synthetic processes. As an example, red blood cells that perform very few synthetic reactions yet require significant amounts of NADPH. NADPH plays two critical roles in RBCs. These two roles are related to oxygen transport function of the red blood cell. Oxygen is toxic, and without reduced glutathione, peroxides that are spontaneously formed from molecular oxygen would oxidize the lipid components of the red blood cell membranes. Furthermore, peroxides have a tendency to damage hemoglobin, causing precipitation of the protein, which impairs oxygen carrying capacity. Two enzymes are essential to deal with the peroxides. They are glutathione peroxidase that converts the peroxide to an alcohol using glutathione, and the second one is glutathione reductase that uses NADPH to regenerate the reduced glutathione. The second role of NADPH in red blood cells is associated to hemoglobin. Oxygen tends to oxidize a hemoglobin ion from +2 oxidation state to more stable +3 oxidation state. As a result, leads to the formation of methemoglobin. This is problematic. The +3 state of the heme ion binds oxygen very poorly. Now, NADPH is used to supply the reducing equivalents to methemoglobin reductase, the enzyme which returns a hemoglobin to its +2 oxidation state. The next role is in the case of glucose 6-phosphate dehydrogenase deficiency. This glucose 6-phosphate dehydrogenase deficiency is a very common genetic abnormality that is found in 5 to 10% of the global population. The effect of the deficiency is decreased ability to form NADPH in the red blood cells. Now, malarial parasites live inside the red blood cells. The normal lifespan of RBCs are about 120 days, but may be shortened by a number of mutations, like for example, if there is a glucose 6-phosphate dehydrogenase deficiency, or if you have a sickle cell anemia and thalassemia, etc., causing the death of the malarial parasite that have not reached maturity. In case of glucose 6-phosphate dehydrogenase deficiency, the most common mutations seem to decrease the half-life of the enzyme, that is effective in reducing the lifespan of the cell. So, the erythrocytes in individuals with glucose 6-phosphate dehydrogenase deficiency are asymptomatic unless they are challenged with oxidants. Oxidant exposure, however, causes hemolytic anemia, that is a reduced number of red blood cells due to the lysis of the cells. So, now we understand how significant this pathway is for maintaining the homeostasis and it should be regulated. The entry of glucose 6-phosphate into the pentose phosphate pathway is controlled by cellular concentration of NADPH. Now, this NADPH is a strong inhibitor of glucose 6-phosphate dehydrogenase. So, whenever the NADPH level goes down by the inhibition is relieved and the enzyme is accelerated to produce more NADPH. The synthesis of glucose 6-phosphate dehydrogenase is induced by increased insulin to glucagon ratio after a high carbohydrate meal. Thus, now I hope you are clear with the factors that control the metabolism of glucose by a balance between the process of glycogenesis and glycogenolysis, and also how reducing equivalents and important intermediates are synthesized by the HMP shunt, and how the amount of NADPH regulates various metabolic activities of the cell by controlling the key enzymes of the HMP shunt. So, having seen this module, we will be having a clear idea about how glycogen metabolism is regulated and also how this HEXOSE monophosphate shunt provides reducing equivalents, which is very important for various conditions of the body.